With that, I'll turn it over to you.
Yeah, thanks, Brian. And first, thank you for coming here and not going to Slope Day, choosing this over, uh, I actually went to get a coffee there and I got enticed. Should I come to my own talk or not? But I said, no, this is more important. And I'm going to share what I've done for past 3 years and how my research program evolved, as Brian mentioned, from ESR to other applications. Um, I'm going to talk about these four things, like molecular mixtures, parametric species, protein dynamics. And I'm going to explain in short what does protein dynamics has to do and why it is important nowadays. Key here is a simple message which I want to say is currently, if you see people have biomolecules, people have spectrometers. It's like cars and people. Question is, where does computation play a role? Actually, till now it was theory, like how can we predict things? But with data analysis, you can actually empower spectrometers, which provide you all the information necessary and take a lot of load out of spectrometers and do that thing without engaging in high software or spectrometer development. So let's start with molecular mixtures first. This talk is divided into four parts. First part is molecular mixtures. And molecular mixtures are important for drug development, food industry, natural products, and chemical industry. And they are part of our day-to-day life. When I started this developing this algorithm, I talked to a few people in pharma. They say, actually, we cannot detect many molecules. When we are at best can do 1 or 2, 3 molecules. And I said, okay, so how can we actually have a mixture and find individual molecules in a mixture? That is not a, I never realized that's a big problem in the industry. And there are several methods to do so, but one has one advantage over the other. This is an NMR spectrum of it. So this is an NMR spectrum of an overlap spectrum. So how do we do molecular mixtures? We actually use these NMR-based methods. One is 2D NMR, pure shift NMR, pseudo 2D NMR, to see can we separate individual molecules in molecular mixtures. Each of them have one problem. Pure shift actually can do a really good job, but it has a sensitivity problem. 2D NMR has high acquisition time. If you want to do high throughput analysis, you cannot do with 2D NMR. And pseudo 2D NMR depends on your molecular size, so it's not generalizable. There are other methods like mass spectroscopy, which sort of burns the sample, and other methods, but everyone has one limitation or the other. There are other computational methods too, say like deconvolution and deep learning methods. The problem is they require a priori knowledge. If you have 100 molecules, you need to know what those 100 molecules are, or at least have some idea of it. Deep learning can only do the things which you already know. So it cannot predict. It can have some predictive value, but not much. And if you have a novel molecule, and that's the key here, if you're looking for something new, deep learning only requires what you have already in the training. So how to overcome this problem? Going back to the basic NMR. What happens is we have a molecule, we do a simple NMR experiment, we get an expected spectrum. This spectrum is what we want. This spectrum, that is what we get. It's an expectation versus reality problem. First of all, there's a noise which suppresses the information. Second of all, there is a resolution problem that you get multiplets. It's overlapped and it's a crowded spectrum. So the question is, if you have a molecular mixture and more molecules come, the spectrum gets much more crowded. How can we solve this problem? Obama is not very happy. Michelle is. She says, well, you can use computational methods to solve these problems. So what sort of computational I want to talk about? Bit of history. It was developed in Germany by this guy Haar. He said, signal is not a signal. If you see back in, say, 70 years ago, you will print the data, right, on a paper. All you can study is x-axis and y-axis. That's it. But signal is a collection of signals. So can we peel that information out into its distinct components, and we can study them? So he first developed a theory, says that can we separate the individual signals which are composed, making these overlap signals? So what does it mean is, so if I take my image, pass it to a prism, what we can get into different colors. We can divide into different colors. This is akin to taking Fourier transform. It divides into different frequency components. But it doesn't tell me where the red is lying in my body. It just tells me I have red. I have more component of red, I have less component of green. But in his theory, he said we can actually differentiate it. Till now, we are finding whether we can see red. Now we can see where is that red occurring, where is that green occurring. So if you look at my picture, you can identify that blue is not probably the best way, and each pixel location will give you a different information. It's not uniform. So that's called two-parametric representation, and that's where the multidimensional aspect comes to picture, where things are occurring and how they are changing, and that can reveal us how to separate the two distinct signals, because every signal will have a different change or different signature, and using these, how they are changing the magnitude and how they are changing in terms of their features, we can predict this part. So what we do is we take there's a Fourier spectrum of a data. We can divide into different frequency bands, which makes the Fourier spectrum, and see how the signal is looking in those frequency bands. So let us go there. So this is an input signal. These are the different frequency bands. Collectively, they form this band. So if you divide the signal into two bands, low frequency and high frequency, and then project how this signal looks in high frequency, you can see these new changes. Similarly, you can see a lower resolution of that image. Then you take this low resolution, divide into two parts, high resolution, low frequency, and high frequency, and then further project it and repeat the process. What it gives you now, this one signal has been divided into 6 signals into its constituent components, and you can go on for a multiple steps. And so you start peeling out information one by one, what it is composed of. Sometimes you need these two together to make a one signal. Sometimes they represent independent of certain feature. And the good thing about this process is that every of these components are centered around 0. So sometimes you need to know what it is, but what to replace these values with. So when it's centered around 0, 0 is the natural occurrence of value. So in the two-parametric representation, Haar in 1909 developed this function which can allow you to probe a particular signal based on how does it change. He did not become very popular. As you can imagine, in 1909, Albert Einstein is publishing all these fancy papers. Who cares about wavelets back then? Plus, there was another problem. There was no computers. So you have a theory which cannot be implemented. Einstein's theory was also not implemented, but the thing is, you can at least imagine that. Then this guy, Morlet, came into the picture in mid-1970s, where the actual signal processing started getting developed. Remember oil crisis in 1970s? So he wanted to find oil. So if you put the signal, seismic data, you're going to get all the noise. So he found this function. He said, let me use this function to predict if they match together. And if they match, we can find the oil. And the strength of this signal will reveal me whether I have how much oil I have. So that's how he developed this. But there's a problem. Anything that does not match with this signal will not be revealed. It can be something else. Signal can be something else too. It doesn't have to be in certain form. So it's a mathematical expression. Then Dobschi's came into the picture. So Dobschi's is a famous mathematician. In 1980s and 1990s, she said, I don't need mathematical form. Suppose I take a picture of mine. There's no mathematical form of my face, but I still have features. I still have magnitude. So the question is what to do about it. So she said, use fractals. Fractals don't have functional form, but they can represent features. They represent that, okay, there's a certain feature and we can extract it. And these fractals were developed based on time frequency analysis, how the signal location changes and how fast they change. That's what I mean, time frequency. And when she developed this thing, this became very general. You can apply on any signal. And I'm going to talk more about it. And this is used in image processing. So everybody now has a camera, right? That is all wavelets and uses her mathematics. Previously, the Kodak cameras used to take negatives. You're going to make the whole picture. Now, using this formula, you can actually take any picture. You can zoom in, zoom out, crop. This is because of this feature. And then when somebody in America develops, French are never behind. They say, we want to pitch in too. So they developed a practical way of implementing wavelet transform. Those who have implemented wavelet transform in 1990s, it was very computationally expensive. You don't have hardware. But he divided a smart way of developing a wavelet transfer where number input data is equal to output data. That's the key. If you have 100 data points, output should also be 100 data points. If you take more data points, then you have a storage problem. and a computation problem back then. And this is where, if you ever watch a video, this is why it is happening. Old televisions used to have this antenna-based Fourier transform base, which will have a receptor. Wavelets are used in all the video processing. So when you do the higher definition and a lower definition resolution, it's all wavelets, where you're choosing a different component of wavelet to show your image. So if you see smartphone has a very low resolution, and the big TV will have a high resolution. And that's what, because Dobishes developed that wavelet, and Madet showed how we can transmit the video at a very low bandwidth. More information can be found in these two lecture books, which are very comprehensive. And we just to tell the contribution of Dobishes, New York Times had a really nice article on her magazine, Godmother of Digital Image. So anytime you see a video and image, it is because of her, because she developed a wavelet, which is not a functional form. So we develop wavelet transforms without going into too much math, just to show that we have been developing fundamental wavelet methods. And you can see all the theorems and lemmas that we publish, but that is not immediately relevant to our talk, but I just want to go through that. We do fundamental wavelet theories. So coming back to NMR. So when you take an NMR spectrum, look at this molecule. One of the basic question is, how many singlets are there? So a lot of times, due to small chemical shifts, a single component can have multiplets, and they belong to same feature. The question is, can we identify those features, single features, and represent them differently? So we use wavelet transform to separate them, because you can see this is a very crowded spectrum, and you don't know what to do with it. So if you apply wavelet theory, you can actually find there are 3 singlets associated with different chemical shifts, not associated with a single element. So how does it work? You take this spectrum, take wavelet transforms. You can see there are multiple wavelet components. Each wavelet component, here you can see it's mostly noise, mostly noise, mostly noise. And suddenly you start seeing in this feature, you start seeing these peaks coming out of this crowded peak. To zoom in, you can see this is a multiplet. You go into next level, again a multiplet. Go to next level, it sort of runs into an enveloping shape. And finally, we get a very single peak, single spectrum. And this peak becomes a singlet. So now we have converted multiplet into a singlet. So those wiggles are basically these things, which is sort of resonant, is converted into a single peak. Now we can separate those features out, and these are wiggles. The peak heights are the most important part of it. Now let us take a spectrum. All 3 of them, these are the NMR spectrum of all 3 of them. Apply wavelets, you get these singlets. And once you get these singlets, you can keep them and discard them. And when you look at the reference, mix them. So in this previous case, these are the individual singlets spectrum with them. Go to the next step, you merge all 3, fructose, sucrose, cellulose, you merge all of them and you get this crowded spectrum. Now, how do we get individual molecules out of it? Apply wavelet shift spectrum, you start getting these singlets. And each singlets now correspond to different component, different molecules, because their location is occurring at a different location or different positions. And you also found some unresolved. So it gives you what is there and what potentially we don't know what there can be additional information. So we can separate the 3 of them, knowing that, okay, once singlets allows us to separate different components into individual molecules. So here you can see the blue, pink, and magenta. They all refer to different molecules. And without wavelet packet transform, you could not have differentiated. It would have been an overlapped spectrum. Similarly, then we do the mixture of 5 molecules and 9 molecules. This is the crowded spectrum. Apply a wavelet packet transform, you get this singlet. And because you already know what those individual molecules look like, you can actually find it, okay, this means this, that means that, and separate them. So these color coding reflects different molecules in a molecular mixtures. Go to 9 molecules, mix them up, get this spectrum. And then apply a wavelet packet transform, the procedure which I described, you can get individual peaks And now you can compare which peaks belong to which molecules in a spectrum. This, yeah.
The lower spectrum, taking the separate spectra of those 9 things and seeing where they show up.
Yeah. Yes. Yes. Now, good question, because the next slide is, can we automate this process? Because last thing you want is all these singlets and somebody really picking out which location where it is. That's what the next step was. We have an unknown molecular mixture, convert into wavelet packet spectrum, and have an NMR database of the wavelet packet cluster of different molecules, and then do a similarity matching, so that any molecule entered there, you can identify this is molecule number 1, molecule number 3, molecule number 4. So how to go about it? This is an NMR spectrum. Apply a wavelet shifts transform, so you get individual peaks. This is without finding the maximum peak height, so you get a wavelet transform spectrum of it. Now, what you do, you already have a library of all the potential molecules. You have a matrix, and then you apply a gradient-based optimization, which gives you a first list of potential candidates. Then you apply the same procedure on this one independently on this 15, and then it finally gives you, okay, this is lactic acid, citric acid, caffeine. So it allows you to separate individual components using an automated procedure. In this case, the key here is, how do we do is, we take 1,000 individual molecular spectrum. We make these 1,000 molecular mixtures are from the combinations of 75 individual molecules. So you have a library of 75 individual molecules. We mix them in, sometimes 3 of them, sometimes 7 of them randomly, generate 1,000 spectrums, and then find out whether we can identify whether 3 molecules are there, 5 molecules are there, or 7 molecules are there, based on those 1,000 library. And what we found is, we actually have a very good true positive rate. So this is the thing with other methods. They have their true positive rate is not that much. So they can only identify, say, 3/4 of the molecules present. If you have 10 molecules, they will say 8 molecules were present, or 7 molecules were present. They are going to miss those molecules. In our case, our median rate is 1. We can identify all 10 molecules. But we do have some false positive rate. Suppose you have 10 molecules, it is going to identify all the 10 molecules, but it may also identify 11th molecule or a 12th molecule. That's the false positive rate. So the good news is, we can get whatever is present. We may get something additional, and then we can actually go back and see which one is the additional. So we do have, but the thing is, we are not missing out on anything. We actually get whatever is present in the molecular mixtures. So this is from the 1,000 molecular mixture spectrum. And we can go up to 7 molecules. If you have a molecular mixture of 7, we can do so. With some effort, we can go to 10, but mostly 7. We can predict that. So this is, you can see this NMR spectrum, and we identified different number of molecules without any a priori training or any a priori knowledge. So the next step is, go to up to 10,000 molecular mixtures. Hopefully expand the number of molecules we can separate out automatically. Currently, we can do 15, then go to 15, 50, or 100. And high field, this was a 500 megahertz magnet. Can we go to a benchtop magnet, 81 megahertz? Luckily, I think Mike Linsky found a magnet for us, and we are working on that part. Just benchtop magnet, put your molecule, and it can spill out how many individual molecules do you have within it. But where I am heading, this is for other people. So again, thanks to Cornell police, they found that cocaine, heroin, cannabis are ample at Cornell police station. They are contaminated by other molecules, and they want to develop a digital forensics lab and create a digital library of all the molecules that you want. So we are collaborating with Cornell police, where they are going to supply us all these molecules. So if you find men in guns having cocaine in their hand, walking in the department, they are here for do science, not to arrest someone. So, and this is where I'm very excited about. It is for 2 reasons. Cannabis being legalized is a big problem. Quality control among farmers and pharmacy is a big problem. So, and there is no regulatory and digital forensics library. And that is what my biggest focus is. Second biggest focus is all the cannabis, as surprising as it may sound. You can go to a dispensary and buy cannabis, but you cannot do research on cannabis. It's a federally banned substance. So you have to go a special farm in Mississippi, which is going to provide you that substance. And that plant does not have genetic diversity. What is going on in the country? So police stations have, they are the ones. And thanks to the parents of undergraduate students who funded all the research, and we can actually find. So first time we were going to study that what sort of diversity all these substances are there, both on campus, and how we can make use of it in terms of identify what the molecular mixtures of them are.
Very interesting. That's also somewhat slightly less nefarious application, that is to the infinite variety of and muscle enhancers, other drugs which affect athletic use. Supposing there is an infinite, we know that organic chemists have an infinite capacity to make any molecule into the sun. So they're making in laboratory some derivatives that have not yet entered commerce and that then have value as, let's say, increasing athletic performance. If you encounter a molecule that is not in any list, you will see it in a gray area.
Yes.
So the advantage you have is for the most, at least for the most dominant molecules, if I understand what
Yes.
you said, the gray area, you have their NMR spectrum.
Yes. Yes, and that's what I think you made a very
So you have a way possibly of identifying those things. But in addition, you could imagine coupling this to theoretical chemistry and generating a database theoretically of derivatives using machine learning. The theoretical basis of molecules that have not yet been made, but that some company in some province of China is busy making. And you could, are the calculations, what I'm really pulling up is the question, are the calculations of NMR good enough today to be able to predict easily an NMR spectrum?
Answer to that question is it depends on the field. So if you take 500 megahertz or slightly higher field, yes, they will have the enough sensitivity. The only problem they had was the multiplets. If you can overcome the multiplets problem, which we try to do so. If you go to a lower field, resolution may be an issue, but we have a high enough field, we can actually do the sensitivity analysis. I think NMR would be good enough. At this.
I think the question has to do with how good is the ab initio theory.
Yes, it's not your work exactly, but if you could couple your work to calculations, you could beat the drug producers in that laboratory and see by not only computing what they possibly mixed, but also computing what they possibly could have made.
Yes. That is actually a very interesting point. I never thought in terms of the gray part. I was only thinking in the remaining part of it. So now coming to the parametric species part of it. So ESR, I think some of you may already use it. It's good at detecting parametric species. So unlike NMR, which gives you everything, ESR can give you a very specific information, which until now may not have been a good idea, but when you need more specific information, it actually becomes a very potent tool. So it is used in, say, life sciences, studying DNA structure, membranes, chemical industry for free radicals or any impurities that we can find. So before I go into the details, it is understand that it gives you certain values. Unlike NMR, which is slightly more better understood, ESR is G values and there are hyperfine constants. And then we relate certain aspects of spectrum or say dynamics or impurity using these values. So let's go here. So in this case, we have systems where we have, say, parametric tag, natural, in this case, copper, or we can simply attach it. We can attach a spin label to it. There are multiple compounds from copper to oxygen, which can be act as a parametric tag. And when we have the spectrometer, the challenge again is similar. First of all, when you run an ESR experiment, you want everything, you get everything, but it is suppressed by noise. In sometimes it is also suppressed by weaker signals. You can only see these features, right? Are there more features to it or not at a smaller number of population? So in this case, I want to give an example. This is an ESR spectrum. This is talking about copper and nitrogen. And we have these G values and A values associated with this copper and nitrogen for the electronic structure. What currently people do is they use simulation methods. So you have this really nice mathematical formulation. And when you get this spectrum, you sort of fit them. You change those values and try to fit what G and A values will give you a better fit of it. These are the simulation softwares. But the challenges have been, you need some a priori information. What are those G and A values? You need to certainly predict. You cannot run through whole spectrum of it. Then it costs more computation. You again have to have a parameter initiation. Sometimes 15, sometimes 2. You need to know what is going on. And more importantly, if your experimental data is artifact, how do you fit it? If you fit to the actual spectrum, then you're also fitting the artifact. And if you're not, then your fit will be poor. So those choices become very important. And as I mentioned, noise itself further adds to the complexity. More the noise, less good fit these simulation methods are going to give. There are other ways of doing things too, quantum mechanics and molecular mechanics optimization. And both they are actually highly accurate, but they comes with high computational cost, and sometimes they're not readily available. So what do? You apply a wavelet transform. This is a wavelet transform. Oh, this is an input signal. You take a wavelet transform, you can now see these hyperfine lines getting separated here. And this structure separated somewhere else. So you can see one component representing this structure, another component representing this structure. And now we can find the associated G values with them, which was previously overlapped. And now you can study the interaction of copper with nitrogens. And this is a simulated data. And we already knew what the G perpendicular and G parallel values are. And when we ran it through the wavelet transforms, we can actually separate the two, and you can see it's quite visual. You can see this is super hyperfine, this is hyperfine, and you can find these values, including these. These looks miniature, but they have a resonance in here. So you can find the G values between them. Now, this is a practical case. That was a simulated case. So we have a collaborator, Arnab Datta. He's a bioorganic chemist. He had a problem that he's studying energy production and oxygen reduction and water oxidation. His problem was he got this spectra and he could hardly see any nitrogen splittings. So we applied wavelet transforms. And I want to go back. You can see this is the thing you're seeing. And after wavelet transform, you start seeing these hyperfine features. The remaining spectra is shown here. So we can choose these wavelet components and reconstruct the data. Now, this is slightly scaled up, but now you can separate the main component with the hyperfine component, which was previously not present, and then use the spacing and the magnitude values to calculate the A perpendicular related to copper, A perpendicular related to nitrogen in this case. And you can also find these peaks and calculate the G values. So this allowed us a really powerful way to separate a signal into its constituent components and reveal the things which a simulation cannot do, because simulation you have to predict it, whether they exist or not. In this case, it immediately shows it. So either it can be used independently, or using these values, you can put it back to the simulation and generate the really nice optimized data.
Let's extract the hyperfine values and independently validate it.
In this case, because we ran through different simulations, so we extracted every value, sorry, in this aspect. So we already know the a priori information. Now, in this case, sometimes we have this what you call validation with this meta, I forgot, where they crystallize the protein or the molecule. And for those which you can crystallize, you can find the counter validation. And otherwise, we see these features and put it back into the simulation, and it should give you the same answer. Because, yeah, that is the. Now, other thing which I do want to extend is these are mathematically generated. So from my perspective, it's much more confident. I'm confident of those equations and getting what information I'm getting. So if there was a junk, it would have given this thing. So yes, so now you can get these values associated with it. Next is with Olga. She's at Syracuse University. Her problem was how many nitrogen ligands were attached with copper center for her hydrogel studies. So in this case, you can see copper mediated formation of hydrogel, and this spectra does not reveal anything. You can see an overlap spectra. Now, when you do a wavelet analysis, you start seeing these features coming in. And then you can calculate all those relevant values, A perpendicular and related to copper, and A perpendicular related to nitrogen. And then you can find out that G perpendicular splitting by copper. And then you can also say copper is coordinated 4 nitrogens. So these features were not readily available to her, but now she can utilize that information and do those things. Another feature of simulation, which is with these methods you can overcome, is sometimes you cannot fit a good second component. Sometimes they will give you a poor fitting because of artifacts. Sometimes it's a good visual fit, but it fails to provide any information in terms of any hyperfine. And these are sort of simulation-based limitations that we can encounter. So simulation is, that's where sometimes even if you give the right G values, they may have these problems because they have not been designed in many cases to do that thing. Now the next question was, can we do a simulation independent? To answer that part of your question, Kyle, we don't know anything about a spectrum. We want to run an independent analysis. So we run a tempo. This is the actual data. Take a wavelet transform of it, you can see start decomposing into its constant components, and suddenly you see one data and the other data coming out of it, and then you separate them. If you go a level before, you can still see there is some sort of hyperfine attached to this part of wavelet component, which needs to be transferred here. So you can immediately separate it. Difference between wavelet packet transform and a discrete wavelet transform that Daubisches developed was, it was to preserve the data length. So that comes as the cost of resolution. So if you're preserving the data length of input data and output data, this does not come at the cost of resolution. It actually is the best way to get a resolution. But you can see that you get so many components. So your data storage becomes big. And that's why wavelet transform has not been applied to, say, cryo-EM with so much enthusiasm, because when GB of data can generate 50 GB of wavelet components, good luck processing that. So
What's the tempo, or is it the tempo attached to something else?
Just of tempo, I believe. Yeah, it's a, I think it's a tempo sample. I think just attached to tempo. I think, wow, I think I know what you're talking about. There's also carbon 13 present in here. This is a small shape of carbon 13, if I am right, if I know right. So this is tempole and tempo. You can go get.
Would be
So we do actually. That is one thing I'm not showing it here.
You're saying coupling to C13.
So this is C13. Yes, these are C13s. These are, I think, nitroxides. Yep. So carbon 13, which is suppressed, we can actually see it here. These are the carbon 13 isotopes. Yep. And now in this case, when you take a wavelet transform, there are multiple wavelets that you can use. And those wavelets, a lot of people think it's like a functional form. So you can give you different answer. Answer is no, it gives you different sensitivity of the data, how you process it. So if you even try different wavelets, say coefficient, Daubisches, you get almost the same answer from there. So it is wavelet independent. So you can simply take a wavelet transform and do that analysis without worrying about whether you chosen a right wavelet or wrong wavelet. These wavelets were generated for different purposes. One is for images, because images have pixels from 0 to 255. So they, as we already know the limitations, so why not have a specific wavelet? Or when you're doing an oil extraction. But the general wavelets can be independent of any data sets. And when you have multiple component, question is that single component is easy. Multiple components becomes complicated because again the spectrum is very much overlapped. In this case, you can see there are two nitrogen. One at 1 to 12.5 Gauss, 116.5 Gauss, which reflects that there are two components rather than a single component. And yeah, and when there's a single component, you get the same values. And then in this case, the future step is if you have multiple single components, you get an overlap data plus noise. Can we get individual with them in an automated fashion? Can we do an automated analysis where if there are multiple components, can we get all these parameters for each component without any issues and see what the error is? So that's what we are currently working on.
What's your intuition about how these work? If I look at on this slide before, we can call the hyperfine spectrum and the super hyperfine
Yes.
spectrum. Calling the hyperfine spectrum looks like a low pass copy of your spectrum, and the super hyperfine looks like your spectrum run through the extreme high pass filter.
Yes.
So is this just a fancy low pass and high pass filter?
It's a low pass and high pass are special case of it. So I would say the other way around. So when you do a low pass filter and high pass filter, these are special cases of wavelets. So in wavelets, you can, because we are doing not just frequency, adding time component to it, so it gives you much more generalized answer. In this case, it looks low pass, but there are some high frequency components to it. If you simply do a low pass filter, it will actually broaden it out. So so it's much more generalized in that aspect. So there is technically only one wavelet to choose from, Daubisches. Now what they did was they said, well, I want a signal I want a wavelet which can maximize whenever I get a signal of feature and minimize when there's a randomness in the data. So they developed a coefficient. Sometimes in MOLLET, they said, I just want to know, I only know what my signal is. Why can't I do a match filtering in that aspect? So they developed wavelet based on that. So, and what this 6 and 9 means is number of coefficients in a wavelet. So if you have few coefficients, your resolution is sort of affected. If you have 6, you get to a perfect sort of wavelet transform. If you go to 9, you overdo it. But the thing is, sometimes depending on how big the data length is, that chooses what your wavelet is. So it's like a filter length in that aspect. Bigger the filter length, better the resolution, but it starts giving a poorer result after a certain length. Now coming to protein dynamics. Proteins are like snakes. They come in all shapes and sizes. And till now, people think that protein is a static feature. So Well, then I actually observed them. I hold snake once, and what I found is actually no. You assume snakes to have all potential orientations, theoretically speaking. Practically, they only have few. So same thing with protein. Protein are highly dynamical, but they have different conformations, and those are limited in nature. So one thing, one advice I gave: when you hold a snake, try to catch from their mouth, because their mouth can really hurt you if you just hold it like that. So I'm going to talk about why long distance protein dynamics matter. Is shorter distances are good, you can find it, but if you want to go to a tertiary structure and see how big changes they are going, you need a longer distance. And X-ray crystallography and electron microscopy can give you pictures, really nice pictures, but then you have to use some modeling, some other approach to find out how they are changing. So we need a certain way to find out whether how big the change is in a much more reliable way. ESR becomes a really potent tool, because what it does, it attaches a two spin label. Either it is naturally occurring, or there are you artificially attached using a side directed spin labeling, and try to measure the distance between those two things. So if you go farther, the mean distance is going to increase. If you go narrower, you can find that. Other aspect of this is, suppose you have 100 molecules, 10 of them have 5 nanometers, but all of them have variant different distances. So then you get a distribution. So the broader distribution, more dynamic the protein. Narrower the one, that means it was very rigid. And what we do is, when you do an ESR, we get this X data. And we want a distance distribution. And as happens with most physical chemistry methods, we never get the data that we want. We always measure it indirectly. So this is the indirect measurement. And this is the thing that we want to include. So this tells us what is the mean distance between those two locations and how broadly they are moving. So it gives you a really localized information, provided that it is aided by electron microscopy, because you need to get the picture of it. How does it look like? And then you can find how does it moves. So this is a pulse sequence of a ESR, pulse dipolar ESR. It's called Deer method, where you apply, in this case, these observation pulse and pulse to get a particular data called echo. And you don't just get the data between two spins, we also get, because there are other molecule too, right? There's not a single molecule between the intermolecular distances too. So you get a background and the interaction between the two molecules, which looks like this. And now you have to generate a distance distribution, which says that I have two conformations with the mean this and somewhat variation. They move in a particular way. And this is a double quantum coordinates, another pulse sequence in pulse dipolar. One has one advantage, another has another advantage, but they mostly give the same information. In double quantum, both the spins are at the same up spin, and the Deer, you have an opposite spins. But I think DQC is much more sensitive to lower concentrations. Deer you can do a lot more experiments with Deer. So let me explain long distance protein dynamics using a special case, Alzheimer's. I hope nobody has it, but that's a disease that is now under a lot of attention. And FDA approved a drug, and it did not work out. I think the PR disaster was really bad for it. And we are still looking for a solution, and a number of patients are expected to grow. So how to solve this problem, and how long distance protein dynamics can help us? One is, this is the schematic diagram where you have a healthy neuron, and when you have an Alzheimer's disease, you start getting this tau aggregates, which is attached to a microtubule. Problem is, it's a disordered protein, intrinsically disordered protein, which means it does not have any shape like a snake. And when it gets aggregated, it actually converts into a shape. We don't know how this moved to here. A lot of times when you do a cryo-EM and other experiments, you get this one line and different sorts of lines like you're seeing it, and you cannot correlate. But there's an underlying assumption, or I'll say good estimate, that actually, although they are IDPs, they do have some partial structures within it. Like the snakes. Snakes can potentially have all kinds of orientation, but they only form a certain orientation. And one of those orientations can be a disease related. So cryo-EM gives you a global analysis, gives the overall picture of tau. X-ray crystallography can give you specific, because X-ray crystallography, you have to have a special preparation. It gives you a certain orientation. And NMR gives you a crowded information. It gives you everything which you need to decouple. ESR gives you a targeted information where you can find distance, long distance between the two spin labels. So what's the problem? We want a distance distribution, we get this dipolar signal. So how to get this distance distribution? We generate a kernel which contains all the potential orientations between the two spin labels and contains all the information about this dipolar signal, and multiply with this distribution, you get that input signal. The problem is we get the dipolar signal and need to find the distance distribution, which is an ill-posed problem. And if you have a much more oscillation, you can get a really nice distance distribution. But if the oscillation is longer, if you have to go collect for longer duration, it's an experimentally difficult task, because longer you go, more time it takes, and there are not enough oscillations to get a reliable distance distribution. So how to solve this problem mathematically? And that is what we have been developing. To get into tau and to have partially structures in a broad distribution, we need two conditions to be met. We should be able to recover broad and narrow features at the same time, and we should have a detailed distance distribution. People use model fitting, but they are limited because IDPs does not have any shape. If you use a Gaussian model, you cannot have a shape. If you use a training-based neural network method, the problem is you cannot train for something that there's no training set available for. And if you use regularization, the problem is you can only extract broad or narrow. You cannot do both at the same time. And that is where we developed our method. And the reason why Ticknov does a bad job is try to optimize the whole distribution at once. So sometimes this point is optimized, this is not. So there's no global optimal point at which you can use regulation and get a perfect answer. So what we did was, we did pointwise. We need a pointwise optimization. This optimization point should not be affecting this optimization point. If it is affecting, we may not have a best solution. So the idea is optimize for each of them independently, and we get a well-resolved picture. Same as images. If you play pixel by pixel, you have a better contrast. So this is a inverse method that we developed, where rather than optimizing for everything else, we just find an optimization for each of them. And we generated a mathematical formula which can allow us to process this data using singular value decomposition. The good thing about singular value decomposition is you can peel it out. You can peel the data into constituent components and reconstruct for each location the way we want to. And I can talk more about it because there are too many mathematical equations in here. So let's take an example of it. This is a ruler data, which says I have a very rigid sample for 2.8 nanometer. This is my collected data from ESR. If I apply a distance distribution, I can easily get this ruler data. Point-wise optimization. And you can also see the uncertainty varies point-wise. However, if I apply the regularization-based method, you can all kinds of broad or narrow. So it sort of misinforms me whether my ruler is broad or narrow, whether it's a dynamic or not dynamic. But it does give you the right mean. So if you are looking for a mean information, it does a good job. But if you're trying to find how dynamical this protein is or this biomolecule is, it's not a reliable method. Similarly, if you do a broad population, you can see in this case, with different evolution time, you can actually find the broad population. And how do we know it is correct? It's because of the PEG. PEG is an intrinsically disordered polymer, and it does not have any shape. So we can actually find whether it's a broad, slightly less broad to more broad, we can collect and find those things. And you can see the uncertainty varies for each point. So we don't have to worry about one point is affecting the optimization of another point. And then detecting local structures in broader population. So in this case, you see we got a narrow peak and a broad peak. So we can detect both of them If you use equalization, you can get all kinds of answers. So going for more data, showing this Brian's collaboration, Brian Crane's group, where we are trying to study how the structural evolution happens. As you can see from one orientation to another, the distance distribution changes, and this peak starts revealing when it is happening, when it is not happening. And we can see those conformation changes with this localized approach. Similarly, with Kevin Gardner, when you activate a light and deactivate, the conformation changes. The biggest problem was you just see a broad distribution, and he wanted a population between two conformations. We know there are two conformations based on his theory. The thing is, this is 50%. This changes to, when you activate in a light mode, it changes to a different population. Height changes. So those are the minute informations. You can actually find it. And that leads to the next point. This is 1D. Can we extend to 2D? And because we need to do protein kinetics, protein folding, and distance assignment. So spin labels you can attach at two locations. Can we actually use more spin labels? Or so distance assignment becomes an important part. This is with Marius Claw group. Because of point-wise optimization and SVD, we can actually extend it to 2 dimension, and you can observe these changes happening. So this is not changing, this is changing. So you can see a protein evolving into a different distribution. And you can again see, you can see that peak height changing. So you can observe how does it evolve with different evolution time. Where I am more focused on this is with collaborators. ALS detection, where ALS unfortunately does not have any diagnostic mechanism. And Stephen Hawking had it. But the key here is all these methods are process of elimination. You first detect it, so you don't have Alzheimer's, you don't have Parkinson's, you don't have XYZ, you may be having ALS. There is no direct method of detection. And the misdiagnosis is about 50%, and it costs up to 40K. So can we actually have a good method for detecting it? Luckily, we got a copper pair embedded in a biomolecule, SOD, which is found in blood. So if we can detect the distance between the two, it can actually inform us what is a disease state, which is a normal state. And this was actually initially work done by Brian, where we can have, there are two distributions associated with it. So you can see they both have similar means, but the width changes. So we need to have a point-wise optimization to measure that width. And that is what we are working on. So if you see blood samples too, that is coming from Weill Cornell, where we can actually, and this is the usually when you put SOD in blood, you don't get a good signal, you have a noisy signal. We have previously developed a noise reduction algorithm. So this is what we are currently working on. Can we reliably detect ALS using ESR, using this distance measurement? If we can, that would be really nice. Last, I don't know how much time do I have, but I will try to finish it soon. Protein dynamics at time scales, different time scales. Till now, we were discussing distance, how far they can go. Question how fast they can go. Sometimes they can go slow, sometimes they can go fast. There are several methods to detect this region. Microsecond is a sweet spot because that is where all the conformational transition states happen, especially 1 microsecond to 10 microsecond. And that is not yet been observed. This is a 2D LDOR, which is an ESR-based method, is one of the unique methods that can actually look at that range. So you can, if you measure the microsecond, you can actually see these chemical, in this case, off state and on state, how the distance distribution is changing, and you can actually analyze the dynamics, that too at the room temperature. That's the key here. Because when people say dynamics, there's no cryogenic dynamics. Like you move in a room temperature, you have to do it at a room temperature. So that's what we are trying to do. The problem is the signal. How do you measure this crowded data? So we apply 2-dimensional wavelet transforms, which further divide into different wavelet components. This is a structure. If you have this 2D data, you can see it divides into several wavelet components. And then can we peel out individual peaks related to dynamics change? Because how these peaks are changing in magnitude, volume, and at what time they are getting escaped. So in this case, you can see this is an input signal. time domain signal is, if you take a Fourier transform of it, you get a crowded spectrum. But can we peel out individual peaks in a way that we can associate with a particular dynamics? And this is a good example of it. You have these peaks, this is crowded, you cannot see it. But if you start taking wavelet components, you start seeing these components peeling out different peaks. And now we can study those peaks in a way independently and see how they are moving at different time scales. Yes. More about multidimensional data processing. I got invitation from Nature Computational Science to contribute a news and views article. So more I can, if anybody is interested, read about it. And MRIs. So this is the method which we are developing is not limited to one application. So if you are wondering whose brain is this is my brain. So if anyone says I don't have brains, here's the evidence. What it is doing is it is reducing the time. If you ever go to an MR scanner, it may take 1 hour to get an MRI scan. Now we are working with UCSF Medical Center. We can reduce time to up to 20 minutes. And for children, you can do scanning without anesthesia, which is a big problem in thing. Some people joke, you can see this region, how well the denoising does. This is the region at 100% quality at 2 minutes 30 seconds. If you reduce it to by 40 minutes, you start recovering it. But if you do it without denoising wavelet transform, you lose that information. A friend joked, look, this shows that you have lung cancer and liver. Look at this invisible tumor. I said, thank you for your diagnosis. Next project is multi-spin. In ESR, we mentioned about 2 spins, but can we do multi-spin experiments? Because that reduces the number of samples. If we have 14 sites to look at, so this is a project which I once wrote an NIH grant, and one of the reviewers said it's too difficult problem to solve, and to which I said we are not in the business of easy. Who solves easy problems? Has anybody ever said that, oh, my research is great because I solve easy problems? So this is where we are trying to show if this project gets accomplished, we can get long distance dynamics without much sample preparation. 1 or 2 sample preparations will be good. And finally, acknowledgements. These are the collaborators I'm working with from France to Germany. Luckily, I got people who are excited about my science. And this is the research group. I have some graduate students, some postdocs, and and undergraduate students too. And thank you for any questions. This is me. I'm a true Cornellian, did most of the things at Cornell. And this is a Billy Connolly, which I sort of enjoy the quote. Life is a waste of time. Time is a waste of life. Get wasted all the time and you will have time of your life. So yeah, I'm having a good time. So thank you.
Any questions?
Question about the use of the term dynamic in the latter part of your talk. So if I understood right, you were talking about initial in the third part or next to last part of your talk about finding static distribution structures.
Hmm.
So am I right that the actually isn't anything to go time scale information or dynamics associated with that? If I recall that static disorder, would that be
Which said which?
the discussion of distance distribution?
Oh, I see.
If you use the term dynamics to refer to disordered structure.
Yes.
As far as I can tell, without any reference to time scale, not actual dynamics.
Yes, and I think that's a good point because time scale ones have to do at a room temperature. In the distance part, what we want to see is how far they go. That is what we
use the word disorder instead of dynamics.
Yes, I think that will be much more fairer term, but I think biologically speaking, they are disordered proteins which can be confused. But I think yes, I would agree on that disorder or sort of how much breadth they have. Out of 100 molecules, they will have, there's no unique conformation, they have just too broad. So I would.
And then part of the talk that succeeded that you talked about the microsecond protein
Yes.
dynamics, is your analysis of these experiments allowing a time resolution that might not have been possible with other analysis techniques?
You're talking about
Resolution.
you're talking about 2D though. For other, we are not aware. I mean, I may be wrong that this microsecond time exchange based, not many people, there are not many methods, and 2D LDOR is one of those potent methods. We are working with Wayne Hubble, who is at UCLA, and he's studying G protein. He's struggling because he did milliseconds, he can do nanoseconds. Microsecond is the stage where all these transitions happen, and he wants this to use this method because that is the unique way of finding what is happening at the microsecond. So my understanding is there are not many methods who can do a microsecond.
That's referring more to the experimental technique.
Yes.
versus the data analysis.
So in data analysis, the question is when you get the spectrum, question is they are too crowded. And so if you do a 1 microsecond, 1.1 microseconds, you get all these 2D spectrums. How do you correlate that information?
So that's the problem is you reach that range of
That can be computationally, it can be done anywhere. It is not limited to the microsecond. You can then to extent. We are also doing on DNP, which is an NMR based technique, because NMR also has this crowded spectrum. But for us, that is the most buck for the value, because this is the thing that many people don't have access to. So that will make a much more impact if we can show it here, and then of course extend it to other methods.
Well, thank you.